Hello, my name is Russ Tashtaki. I'm a biological sciences tutor here on the first floor of Hill Hall at Winston-Salem State University. Today's lesson is going to be on the cell cycle. Now, the cell cycle is the process of a cell during its life. Now, most cells replicate through a process called mitosis. Meiosis is reserved for egg and sperm or sex cells. All other cells are called somatic cells. So this is an example of a somatic cell during its life and what happens, okay? So the cell cycle itself is in four main phases, and you can see that here, okay? You have the G1 phase, which is the growth phase. The cell is born, it grows, it's young, kind of like somebody who's in college, for example, living their best life, having a good time, enjoying their youth. And then you get older, like me, and you start getting gray and you realize, wow, if I want to reproduce, I better reproduce, okay? So then you copy your DNA, all right? That's done through this phase here, the S phase. See, that's quite a big chunk of the cell cycle. The S phase is called the synthesis phase, where you copy your DNA in a process called DNA replication. So in a previous video, we talked about transcription and translation. Transcription was the process of taking DNA and RNA that happens in the nucleus. Translation is the process of taking that RNA and making a protein on a ribosome. This is called DNA replication, which means copying DNA into DNA. So the A binds with T, the G binds with C. I grew up in Greensboro, North Carolina, and we have two big schools there, A&T State University and Guilford College. So I remember that as A&T and G and C based on those schools. So if that helps you, that's great. So A binds with T, G binds with C. Again, if it's RNA, A binds with U, uracil, and G binds with C. But this is DNA replication. So we're taking DNA to DNA in this phase called the S phase, okay? So the synthesis phase, you're making a copy of that DNA and using an enzyme called DNA polymerase. When you're making RNA, you use RNA polymerase. Remember, ACE is a place for enzymes, ASE. Things that ends in ACE are enzymes, okay? So the ACE, in this case, is DNA polymerase. You copy that DNA, and then the cell lives out its final days, okay? And that happens in G2, which is this phase right here, G2, okay? So after G2, you do this phase called mitosis, the M phase. And it's only a small part of this whole cell cycle. The M phase can be mitosis or meiosis if it's sex cells, egg and sperm. But in this case, it's a somatic cell, which is everything but egg and sperm. So we're talking about mitosis, okay? The phases of mitosis, again, that happen right here. The phases are prophase, metaphase, anaphase, telophase, and cytokinesis. That's where you cleave the cell into two brand new cells. So most of the cell cycle is in what they call interphase. All of this is interphase. Once you get here, then you do the prophase, metaphase, anaphase, telophase, cytokinesis of mitosis, okay? So remember, most of the cell's life is spent in interphase. Now, once you go through the steps of mitosis, you end up with two brand new cells. And the biggest phases that are really most important are metaphase, where all the DNA lines up in the center of the cell, and then anaphase, where the DNA starts to split into opposite ends of the cell. And if you remember my video on the cell, we talked about the centrioles, centrosomes. They move the DNA to opposite ends of the cell. They're like little DNA magnets, and their goal is to pull that DNA to opposite ends of the cell so that when you cleave it, you're not cutting any DNA. So you have identical copies of all of that DNA on either end of the cell. And then you do telophase and cytokinesis, where you have that cleavage flow where you're cleaving or you're cutting that cell into two new cells. And at that point, you got two brand new cells with identical copies of your DNA, and then you go back into G1 again, and then S, and then G2, and you do this all over again, except for certain cells, like brain cells, for example. Those cells do this once, and then for the rest of your life, they don't multiply anymore. So that's why if you fry your brain doing drugs, which I hope none of you are doing, you damage those cells, and you can permanently damage them, and if they die, you don't get them back. Now, nerves like in your arms and legs, they're myelinated nerves. Myelin is the insulation that's wrapped around the nerve. Those nerves do grow back because the insulation, the myelin grows back together, and then the nerve follows that path, that insulation. It would take months, but it does grow back together. So heaven forbid, if you cut a nerve in your arm, for example, it will grow back. So if this is a brain cell, it ends up in something called G0, where you arrest that cell cycle. You basically stop this whole process. So this doesn't happen anymore, okay? So when you end up in G0, you hit this checkpoint, okay? That basically says, hey, no more mitosis. So once you get to that checkpoint and it's done, and it's like, well, we're not going to multiply anymore, then that's it. You have that one cell, like a brain cell, that when it dies, that's it. It's not going to copy itself before it dies. So only certain cells stay in G0. But most cells, once they go through mitosis, the prophase, metaphase, anaphase, telophase, cytokinesis, will end up going back through G1, S, G2, and back into the M phase, which is the mitosis phase, or meiosis if it's egg sperm. So I hope I helped you understand this cell cycle. Again, my name is Russ Sashaki. I'm a biological sciences tutor here at Winston-Salem State University. You can find me on the first floor of Hill Hall, and I'll be glad to help you. Have a nice day.